National test. Oyo masami seka webia. He is a the managing director. This electrical merchandise. Apple one of President there. Oyo mabulu mwe nanya. It's always there. No idea. People's Club of Nigeria. People's Club of People's Club of Nigeria. People's Club National President, please try to secure me, okay? National President, National President, Sobele. Iji Kaloba, Agaja Kubia, Sloye Highness, this year's a man, as a Boko one of Ono Mekanekio, Ugena Lego, Busto. National legal advisor, measure one of a jojo, he can knock the zone is so near. Chikelo, Zapupe, Putama Pagisa. She could be Olive, I could not forget you, Yapmania. National law, someone for your Pagaso. It's just like it's on a beautiful love on it. I believe she goes to love. Because you put, I can't let the low, I will go no man is so big. They say you go forget it, you have my anniversary. It's a royal highness, Urubaka, royal highness, Urubaka. It's all one of for it. Urubaka to the worship. What come on the zoo? Back to the worship. What come on the on your Urubaka. Michael Abalasopio, the champion of the champions. Oh, Michael Dobu. No machine anywhere, this over the pair. Let's go one of our repeated. Malam, in what you Sunanke the daddy. Abagid so. Jebolingo. Oh, no man ever win our one in there. What is it to see? See your cake. Oh, no making it. No compo, there's a program of a lot of she see more of your what's one now, Baron this of Noma. But it's a lawyer, I no money to solve. Yes, you're not so well. Worry me. Asinaga madero veleno. Emasiobi. Unyomani sobele. Unubesile pesino. Ha. Oliva lemsekio. Makibia chibumile tue. Oliva lemsekio. Makibia chibumile tue. Robo motivia manyano. Nukumono subeda waloso. Isi rabuna no namadu kidia. Agwani neti kulo kumanya nleke mobia. Adagwa chiti na desu. Namwa malo ni ya malo condition noa. Angwalile no ti abrosinga. Ile kibia. Oliver Nemsekio, oh Lulelo. Rivers to love, baby, I can make you. Oh, the Vizanda, money, I can make you. Rivers to love, I would not say. Oh, the Vizanda, money, I can make you. Oh, baby, I can make you. so, until I come in there, you can eat so delay. What's the club of Keke? Yes, I'm a mother. I feel like a good one is one name. I got my ladder of Helen. Dr. Chukumoga. No money so billy. People's club for Desulua. For Tubulu Mari Banana. I'm well for number. For the Nina decision and Nigerian. People's club for Desulua. Unu Basile Kasi. Abono do so. Jenny Lane. Each one of Bobunike. One is the chisel. Kibu Jenny Lane. Each one of Bobunike. Kibu's full of Bodo. Samuel Manelli. I can let the leader. Masus Biao. Ibo. Let's man dog. Let's man dog.
Good afternoon, distinguished ladies and gentlemen. Please, we have a few minutes to actually commence this very important inaugural lecture. I want to plead with us, if you are in this auditorium this afternoon, to please have your seat. Kindly take your seat. We have a few minutes left to the commencement of the 62nd Inaugural Lecture. Please, let's have a seat. It's very, very important. I'm very much particular about the entrance to this auditorium. Let's make it as clear as possible. For us in this university, you will agree with me that inaugural lecture is not a cocktail party. It's not a champagne party. It's a solemn academic exercise that is given the much needed decorum. Therefore, I want to plead with you. Let there be restriction of movements at this juncture, preparatory to the commencement, honestly, of the 62nd inaugural lecture. Sericom members, please. And if you are in this auditorium, it is imperative and of course expedient of you to strictly adhere to COVID-19 protocols. Your face masks should be on, and social distancing should be maintained. Like we used to say, COVID-19 is not scamdemic, it's pandemic. Passes the Zoom, please. Let's clear the inroads to this auditorium. Let there be silence, please. Thank you so much for that. I'm still not comfortable with the entrance leading to this auditorium. I'm not comfortable with that place. I think it's better now. Thank you so much there. We have a few minutes left to the commencement of the 62nd Inaugural Lecture of our great university, Federal University of Agriculture, Biography. Please, just before we formally kickstart this program, permit me to humbly inform this gathering that there are rules, and I repeat, there are rules guiding the conduct of this inaugural lecture. And I want to plead with you passionately to kindly adhere strictly to these rules. Just like I mentioned earlier, for us in FUNAAB, we believe deeply in decorum. And if that should be exhibited, it should be at an inaugural lecture of this nature, which is purely, to an appreciable extent, academics. So let there be decorum. Movement should be restricted right now. Thank you so much. This afternoon, by the special grace of God, our university has indeed come of age, and we are pleased to witness, to the glory of God, the second inaugural lecture, the 62nd inaugural lecture of our university, and it's going to be delivered by no other person than Professor Christian Obiora Undubuse IKEOBI. He's our guest lecturer today. He's our inaugural lecturer. He is a professor of animal genetics. And very soon, we shall be kickstarting this program. But in the meantime, I welcome you on board of our 62nd Inaugural Lecture. Kindly fasten your seatbelts as the flight of our inaugural lecture is about to take off. Today, we are dissecting a thematic concern. We are dissecting a topic that I believe is quite germane towards development of every facet of our national lives. We are dissecting the topic why we look at things not seen. Animal genes and their winking eyes. I take it again. Why we look at things not seen, animal genes and their winking eyes. Since I've been seen at this particular title, one aspect of the title has been of much concern to me, and eagerly I've been looking forward to it, and that is animal winking eyes. Animal winking eyes. Well, okay, right now, shall we all stand for the academic procession? Let's rise for the academic procession right now. Let's remain standing for the academic procession. It's yet another historical event in the annals of history of our university. The 62nd Inaugural Lecture of the Federal University of Agricultural Abeokuta, FUNAAB. Right now, coming in referently is the much needed academic procession for this occasion. The procession is still in progress, distinguished ladies and gentlemen. Yes, I see principal officers and the professor just coming in. The university librarian, Dr. Mrs. Onifade, the bursar, Mr. Chukwuike Ezepiasu, and of course, the registrar, Dr. Akimbola Adekola, the Deputy Vice Chancellor of Academics, Professor Mrs. Bolanle Akedodo-Ale, and of course, the Deputy Vice Chancellor of Development, Professor Clemens Olabinjo Adeofun, and of course, the inaugural lecturer, Professor Christian Obiora-Ndubusi, IKEOBI, and of course, the Vice Chancellor, Professor Felix Kolawole Salako, who is the chairman of this inaugural lecture. Please, let's give them a round of applause as they are taking their seat this afternoon. You are most welcome, sir. While we remain standing, we take, first and foremost, the national anthem, and thereafter, we'll take the universal anthem. Right now, the national anthem. FUNAAB anthem. A round of applause, please. Please. Let's have a seat, please. FUNAAB anthem always inspires me. It's quite revealing and quite thought-provoking. I want to welcome you all specially to the 62nd Inaugural Lecture of our great university. But please, before we proceed, I told us earlier on that there are rules guiding the conduct of this inaugural lecture, because it's not a cocktail party, it's not a champagne party, but more importantly, it's a solemn academic exercise. Therefore, let us reduce restriction of movement. I will appreciate if you are staying glued to your seats this afternoon. And again too, I want to plead with us, let's strictly adhere to COVID-19 protocols. Thirdly, I plead with you passionately, put your mobile phone on vibration, on perfect silence this afternoon. Let's give the much needed decorum to this program. Thank you so much. I see some people moving around. Please, let's curtail that. We must respect this program. Ceremonial committee members, please. Thank you so much and God bless. At this juncture, distinguished ladies and gentlemen, we have with us on this side of the auditorium this afternoon, presiding over the affairs of the 62nd Inaugural Lecture of our university. We have with us the Vice Chancellor of our university, Professor Felix Kolawole Salako. You are most welcome, sir. And of course, the Deputy Vice Chancellor, Academics, Professor Mrs. Bolanle Akeredoluwale. The Deputy Vice Chancellor, Development, Professor Clement Adiofun. You are most welcome, sir. The Registrar, Dr. Bola Adekola. You are welcome, sir. And of course, the Bossa, Mr. Chukwuike Ezepiazo. You are most welcome, sir. And we have this with us as well, the University Librarian, Dr. Mrs. Feintola Onifade. We have with us this afternoon the 62nd Inaugural Lecturer of our University, Professor Christian Obiora Ndubuse IKEOBI. You are most welcome, sir. We have the chairman committees of deans and directors who also double as the deans of Copeland in our midst this afternoon, Professor Murphy Atayeshi. The dean of Kolanim, that is the dean of the College of the Inaugural Lecturer, Professor Olushiji Showande. You are most welcome, sir. Without wasting our time, distinguished ladies and gentlemen, at this juncture, I want to humbly invite our Vice Chancellor, Professor Felix Kolawole Salako, to give us his welcome address. Let's welcome him here with a round of applause.
May I respectfully call on Professor IKEOBI to stand and remain standing. Please come there. The Deputy Vice Chancellor, Academic, Deputy Vice Chancellor, Development, the Registrar, the Bursar, the University Librarian. The inaugural lecturer, Professor Christian Obiora Ndubuse IKEOBI, Dean, College of Animal Science and Livestock Production, Deans of Colleges and Postgraduate School, Directors of Institutes, Centers, and Services, Head of Department of Animal Breeding and Genetics, members of the IKEOBI family, invited guests, members of our great university, gentlemen and ladies of the press, distinguished ladies and gentlemen, and our own great FUNAABites. Great FUNAABites. I'm delighted to welcome you all to the 62nd edition in the series of inaugural lectures of the Federal University of Agriculture, Abeokuta. This inaugural lecture is the ninth I'm presiding over as the Vice Chancellor. The inaugural lecturer of today, Professor Christian Obiora Ndubuse IKEOBI, was born on April 23, 1959 in Urumechi village, Ozubulu, Ekujigo local government area, Anambra State, to the family of late Sir Dennis Chukwuemeka and late Virginia Akoma IKEOBI. Professor IKEOBI had his early education at St. James School, Uga, where he finished with distinction, and secondary school at Dennis Memorial Grammar School, Onitsha, in 1976. He proceeded to the then University of Ife, Ile-Ife, now Obafemi Awolowo University, in 1977. Graduated with BSc Agriculture Animal Science option with second class upper division in 1982. He had his postgraduate training at the University of Ibadan, where he obtained MSc with the best result and an average of 72.85% in the MSc Animal Science graduating class of 1984. He also went to University of Ibadan to have a PhD in animal science and animal breeding and genetics in 1990. Professor IKEOBI began his academic career at the Department of Animal Science, University of Ibadan in 1986 as a graduate assistant. He joined the Federal University of Agriculture, Abeokuta in 1990 as lecturer 2 and has risen to the position of a professor of animal breeding and genetics since 2003. The focus of Professor IKEOBI's research in the area of animal genetics has been on genetic characterization and evaluation of Nigeria's animal resources, with special emphasis on the local chicken and on the identification and mapping of quantitative trait locale affecting broiler chicken traits and disease resistance. His research efforts have resulted in publication of more than 180 articles in learned journals and other outlets. A very effective academic and mentor, Professor IKEOBI has supervised many undergraduate projects and over 60 postgraduate theses at master's and PhD level. He has held several administrative positions, including being Dean, College of Animal Science and Livestock Production between 2011 and 2015. Dean, Student Affairs between 2005 and 2008. Head, Department of Animal Breeding and Genetics between 2009 and 2011. He's a member of the University Senate and has also been a member of the Governing Council of the University from 2014 to 2018. It is worthy of mention that as head of department in 2010, he attracted a grant worth several millions for 8 postgraduate students research and equipment endowment to the department. In addition to the above, Professor IKEOBI has served in notable statutory body and committees in the university, such as being University Auditor since 2004, Chairman, Senate Committee on Examination Results, 2006 to date, Appointments and Promotions Committee, among others. He has also served in numerous standing and ad hoc committees in the university. As a mark of national relevance, Professor IKEOBI has served in no less than 10 other Nigerian universities as external examiner, professorial appointment assessor, as well as in visiting capacity. He is a member of the following professional bodies: Registered Animal Scientist, Nigerian Institute of Animal Science, Fellow, Nigerian Institute of Animal Science, Fellow, Nigerian Society of Animal Production, Fellow, Animal Science Association of Nigeria, Fellow, College of Animal Scientists of Nigeria, Live Member, Nigeria Society for Animal Production, Member, Genetic Society of Nigeria, and is also a Commonwealth Fellow. I want to attest to Professor IKEOBI is a committed Christian with an undeniable spiritual experience and a special for spiritual regeneration being displayed as the chaplain, Chapel of Grace, FUNAAB since 2007. Perhaps this spiritual trait was what St. Mary Anglican Church, Ozubulu, saw and invested Professor IKEOBI along with the wife with the knighthood of St. Christopher in the Anglican Communion on June 27, 2009. Professor IKEOBI is married to Lady Elizabeth Olubanke, and the marriage is blessed with successful children to the glory of God. Distinguished ladies and gentlemen, it's my honor and privilege to invite Professor Christian Obiora Ndubuisi IKEOBI from the Department of Animal Breeding and Genetics to present his inaugural lecture titled Why We Look at Things Not Seen, Animal Genes and Their Winking Eye. Thank you for your attention and God bless you all.
The Vice Chancellor, Federal University of Agriculture, Abeokuta, Professor Felix Kolawole Salako, FSSSN. The Deputy Vice Chancellor, Academic, the Deputy Vice Chancellor, Development, the Registrar, the Bossa, the University Librarian, the Chairman, Committee of Deans and Directors, and Dean, College of Plant Science and Crop Production, the Dean, College of Animal Science and Livestock Production, deans of other colleges, postgraduate school, and student affairs, the directors of centers and directorates here present, the acting head, Department of Animal Breeding and Genetics, other heads of departments and units here present, my lords, spiritual and temporal, members of the Governing Council here present, members of Senate, members of congregation, and other colleagues here present. I would like also to greet the vice chancellors of other universities here present and former vice chancellors of this university and of other universities here present, members of the IKEOBI family, especially my loving wife, Lady Elizabeth IKEOBI, and other invited guests here present, distinguished guests, ladies and gentlemen, great Unabites. It gives me great pleasure to present the 62nd inaugural lecture in this university. And it's also an opportunity for, someone has said, inaugural lectures are an opportunity for one to talk about his research and his endeavors since he was employed as staff of the university, and also an opportunity to showcase one's contributions to both town and gown so that people will understand what the contributions have been. This inaugural lecture is the 11th in the series coming from the College of Animal Science and Livestock Production, and also the 4th from the Department of Animal Breeding and Genetics. I am therefore privileged and blessed to have this opportunity to take this time out with this inaugural lecture. The title of this inaugural lecture is Why We Look at Things Not Seen, Animal Genes and Their Winking Eyes. The inspiration for this title was derived from the Holy Bible in 2 Corinthians chapter 4, verse 18, which reads, why we look not at the things which are seen, but at the things which are not seen. For the things which are seen are temporal, but the things which are not seen are eternal. Genes are the basic units of inheritance and are transmitted from parents to their offspring in all living things. They are not visible to the naked eye, but they are detectable, and they manifest their effects phenotypically in ways that can be observed or measured or assessed or evaluated. Of course, genes are basically located on chromosomes at points that are called loci, and the singular is locus. And there are different types of chromosomes in animals. In man and other mammals, there are autosomes and sex chromosomes, X and Y. But of course, in chickens and other poultry species, there are 6 pairs of large macrochromosomes. And some earlier authors have put it at 8 large macrochromosomes. But in recent times, the consensus has zeroed into 6 pairs of large macrochromosomes in chickens. And there are also 33 pairs of smaller microchromosomes, and there is a pair of sex chromosomes, Z and W. And microchromosomes are more gene dense. In other words, they are richer in genes than macrochromosomes. And there is a 6-fold difference in gene density between microchromosomes and macrochromosomes. And the reason for this is that there is a higher rate of recombination events in microchromosomes vis-a-vis macrochromosomes. It has been estimated that about 50 to 75% of all chicken genes are born on microchromosomes, giving rise, and this of course gives rise with, when you have genes that are born on sex chromosomes, you have traits that are sex-linked. Now, why this background? We have carried out series of researches in which we identified genes and quantitative trait loci in both chicken autosomes and also in sex chromosomes. And we'll be providing details during this lecture. Issues of life, any life, are summed up in these 3 crucial areas of relevance: influence and impact; productivity, which has to do with increase in growth and multiplication; adaptability, which has to do with tolerance and susceptibility to environmental and climatic situations; an ability of the individual animal to thrive and produce and reproduce; and then livability, which has to do with the ability of an animal and offspring to survive. And part of our research has had to do with these areas of relevance: productivity, adaptability, and livability of animals in different conditions, and the genes that have to do with each of these major productivity groupings. People have said that the tropical environment in which we live is characterized generally by stress factors. And these stress factors include high ambient temperatures, high relative humidity, high solar radiation, poor feed resources, poor water availability, and poor management practices. The animal environment is important for us to understand that this is an important component in the animal genotype to phenotype relationship. And of recent, the phenomenon of climate change has had serious impact on animal productivity, adaptability, and livability, and on feed resource availability, quality, and diversity. Livestock rearing, it must be noted, has contributed substantially to this phenomenon of global warming and climate change, as cattle have been implicated through their gas emissions as major contributors to global warming. An adult cow, it has been estimated, emits 70 to 120 kilograms of methane to the atmosphere each year, the Michel 2018. And when we consider the global population of cattle, which has been put at 1.5 billion, we know that the contribution of the global population of cattle to gas emissions, in particular emission of methane to global warming, is substantial. And of course, other greenhouse gases that are implicated in global warming include nitrous oxide, carbon dioxide, and others. And factors affecting gas emission in cattle include feed intake, feed processing, type of carbohydrate in the diet, rumen microflora, and the types of rumen microflora in the diet, in the rumen, environmental temperature, and addition of lipids in the diet. I specifically remember that while we were growing up, as primary school students, as primary school pupils, each time that my grandmother's goat suffers bloat, my grandmother will rush to give a spoon of palm oil to the goat suffering bloat, and within minutes, that situation will be ameliorated. And so the addition of lipids in the diet has been proven in published literature to be one of the factors that can affect gas emissions, and it can also reduce it. Now, livestock production in Nigeria entails the rearing of several livestock species, poultry such as chickens, ducks, turkey, quails, guinea fowl, ostriches, rabbits, pigs, sheep and goats, cattle. And RIM 1991 has previously put the population of chickens to be 120 million, 30 million for goats, 22 million for sheep, and 12 million for cattle. We'll be making some comments along this line during this lecture. My research contributions. Vice Chancellor, sir, my research in the university since I assumed duty in 1990 as a lecturer here in this university has centered on these areas: quantitative genetic characterization and evaluation of the productivity of Nigeria's indigenous animal resources, especially the Nigerian chicken. It has also centered on the identification and mapping of quantitative trait loci, in bracket genes affecting broiler chicken traits, and also on the evaluation of molecular genetic diversity and associated traits in Nigeria's livestock species. And details of the contributions we will try to highlight the major areas. There is no way this kind of lecture will encompass every area, but we will be given excerpts from each area. On the area of quantitative genetic characterization and evaluation of Nigeria's animal genetic resources, at the early part of my career in FUNAAB, we carried out a retrospective study which dwelt on the productive potentials and the occurrence of major genes of frizzling, naked neck, and dwarfism on the chicken, in the chicken. And the result of that study highlighted the unique features and genetic potentials of the Nigerian chicken, which can be exploited in future breeding and improvement programs based on it being a repository and supplier of gene complexes affecting productivity, adaptability, and livability traits in the species. Ebozeje and IKEOBI, 1995. Collaborating with other researchers, we have been able to genetically and comparatively characterize the Nigerian chicken with respect to productivity and the effects of major genes of frizzling and naked neck in both the extensive farmer-managed flocks and in intensive systems in our university farm here in the university. My research and publications extend the base of knowledge available on the roles of major genes, skin and plumage pigmentation genes, and plumage distribution as adaptability traits of body temperature regulation and productivity indices have been affected in our hot, humid environment prevailing in southwestern Nigeria. We've also estimated the frequencies of these genes, in the local environment, and we have also highlighted the need to conserve these genes and the chickens bearing their physical manifestations for the benefits of the present and future generations of humans. IKEOBI et al. 1996 reported that the frizzled local hen laid and hatched the least number of eggs per year under the open range system of management compared to the naked neck and the normal feathered birds under similar conditions. Of course, drawing from that, we also reported that the egg weight significantly favored the frizzled chicken because there is an inverse relationship between egg production and egg weight. And our study brought this out very clearly. We also found out that we also evaluated semen quality of 7 chicken genotypes, Peters et al. 2008A, and semen volume was highest in white leghorn birds, which is an exotic chicken, and least in local naked neck cocks. But the naked neck cock had the highest value for motile spermatozoa, while the Giri Raja, which is an Indian breed of chicken, had the least. We found out that large genetic variation existed in semen quality of cocks, which could be exploited, and indeed is being right now exploited in breeding programs. The least hatchability was obtained with naked neck eggs with the highest percentage of dead in shell, and this confirmed what had been written in literature about the lethality of the naked neck gene when in the homozygous dominant situation. There is a high percentage of dead in shell, and our study brought this out very, very clearly. But when you have the heterozygous naked neck situation, this lethality is reduced substantially. We went on to study the effect of crossbreeding local chickens with an exotic strain on fertility, hatchability, and embryo mortality, Adeleke et al. 2011A. And frizzled sired cross gave the best fertility of 98.5% and the best hatchability, 96.8%, relative to the other crosses. In another study, IKEOBI et al. 1999 found that the eggs of Nigeria's local poultry species, and this also includes ducks and guinea fowls and the Nigerian chicken, they met the prevailing United States standards for quality of industrial eggs as earlier categorized. And this means that the eggs of Nigeria's local poultry species could be used. They met the standards for industrial eggs set by United States, and they could be used for industrial production. And indeed, further research going on in the College of Animal Science and Livestock Production have gone to process the eggs of these chickens into powder, and this could be used for industrial purposes. The research, this later aspect I've mentioned, goes on in the Department of Animal Production and Health. Now, we also conducted a study to investigate the genetic variation in the carcass traits and blood parameters of local naked neck and normal feathered chickens and their crosses with Marshall. In the study, the naked neck and the normal feathered chickens had the lowest values for mean hemoglobin, white blood cell count, red blood cell count, and also had the highest values for globulin. Now, I should also give you the illustration. What you have on the screen is the frizzled feathered local chicken on the right-hand side of the slide. And then on this slide, you have the normal feathered chicken on the left-hand side and the naked neck chicken on the right-hand side. These are things you would have been observing in the local environment from time to time. But these chickens, we have found that indeed have major roles to play in enhancing food production, poultry meat production in Nigeria, because in the villages, these chickens command great attention in terms of meat production, egg production, and chicks production. The other side of our work was the evaluation of rare qualitative characters in local chickens. And we looked, apart from looking at the major genes of frizzling and naked neck, we also looked at rare qualitative characters, polydactyly in the Nigerian chicken, which is controlled by the polydactyly gene, capital PO. The cresting gene, which is controlled by the cresting condition, which is controlled by the cresting gene, CR plus, capital CR plus, and then the tylopody feed feathering condition, which is controlled by a gene PTIL, capital PTIL, and then the comb types, and then brachydactyly. We have this chicken on the right-hand side of the screen, which is the polydactylous local chicken. And polydactyly is the presence of extra toes in the chicken feet. And this is a very, very dominant situation we have found in the Nigerian local chicken. And we are recommending that these chickens be conserved for the benefits of the present population and future population of Nigerians. You have also the cresting gene. What you have on the screen, there is a tuft of feathers on the head of the chicken on the top, and then you have the rose comb, because we looked at comb types in the Nigerian chicken. You have the rose comb. And what we have found with respect to the Nigerian local chicken is that the single comb, which is what we have on this bird that is that has naked neck, the comb type on it is the commonest that we see around. That is the single comb. But the gene for the single comb is small r, small p. That is the gene. There's an interaction of genes to form that situation, that condition. But interestingly, in spite of it being in the recessive state, recessive homozygous state, homozygous recessive state, you've still found that the single comb is the predominant, the most prevalent, indicating that the situation where you have natural selection taking place to ensure this livability for this. And along that line, Siegel and Dudley, 1968, found that these birds with single comb were more agonistic in behavior related to birds with the rose or pea comb. And this must have ensured the survival of and the high prevalence of the birds with single comb in our environment. Further studies on this should be embarked upon to ensure, to confirm that this situation in our local environment. Part of our study also has to do with pigmentation genes. And in one of our studies, we found that the plumage pigmentation, which is genetic in origin, significantly affected the body weight, the breast girth, the breast length, and egg production in the Nigerian chicken. Shank color also significantly affected the weight of the birds. Adebambo et al., 1999. And the significance of the shank color is that oftentimes it has to do with the carcass color, the color of the meat of the bird. And this is very, very important. And that's why we evaluated shank color and their prevalence in this condition in our environment. In one of our studies, we reported on the occurring patterns, mode of inheritance, and frequencies of plumage, skin, beak, and earlobe pigmentation genes in local ducks, local turkeys, guinea fowls, and pigeons. Ozoje et al., 1999. We went on to look at coat pigmentation in goats. And our study, Ebozoje and IKEOBI, 1998, found that the reproductive performance and pre-weaning growth in West African dwarf goats significantly favored goats that were black in color. And we also found that there were several genes controlling coat pigmentation in the West African dwarf goat. And one of those loci is the B locus, which controls the expression of black color. You also have the A locus, which controls the expression of tan and other colors like that, like we have on the right-hand side of the screen, of the plate on the right-hand side. We also have the S locus, which controls the expression of white belly markings on the animal. And what this is saying is this: when you talk about West African dwarf goat, you cannot compare, you cannot do a research oblivious of the coat color and tell us that maybe you put the black goat with the brown goat and the white goat and tell us they are all West African dwarf, because there are specific genes, the A locus, the S locus, and the B locus, that control different expressions of different coat colors in the West African dwarf goat. And this, of course, has to do, because when you talk about color, you are talking about the ability of the animal either to absorb, the environmental solar radiation, or to disperse it. And this, of course, affects body temperature, affects pulse rate, and affects the general performance of the animals. And so this is a very important contribution in our study, showing that different coat pigmentation genes in the same West African dwarf breed, they affect the performance of the goats in different ways. Part of our work also was on direct genetic effects, maternal effects, and genetic parameter estimates. And IKEOBI 1998 reported high heritability estimates for body weight 0.71, egg weight 0.42, and low heritability for egg production. Of course, being a reproduction trait, that's understandable, and our study confirmed that. Very interesting, at least in conformity with theoretical expectations. IKEOBI 1990 and IKEOBI and Ngere 1994 reported that direct genetic effects were important for pre-weaning piglet weight and survival. IKEOBI 1993, and for pre-weaning litter size. This is part of my PhD at the University of Ibadan. And using the allele analysis involving the local chicken and some exotic chicken strains, we found that both additive and dominance gene effects were important in feed efficiency in the local chicken. Feed efficiencies were low for the normal feathered local cocks, 0.12, and normal feathered local hens, 0.14, compared to an exotic hen, Anak Titan, 0.22, Adebambo et al. 2008p. And one of my PhD students, who is now a professor, Professor A.O. Adebambo, worked on this for his PhD. The results of the dialect analysis to test for general and specific combining abilities of breeds of chickens showed that the additive genetic and maternal effects were important for economically important productivity traits, while dominance effects were important for the survival traits. Again, this in conformity with theoretical expectations. Adebambo et al., 2010, 2011, 2012. In line with our findings on local turkeys, IKEOBI et al. 1995 had earlier reported significant sexual dimorphism in 2 strains of commercial broiler chickens. Our studies on modeling showed, we looked at modeling of growth in Nigerian local chickens using Gomper's, monomolecular, and Richards to feed weight age data for 7 chicken genotypes, 4 of which were Nigerian local, that's frizzled, naked neck, and normal feathered involved. And all these models underestimated the asymptotic mature weight of the chicken, but the Gomper's function gave a better or the best estimate compared to the other 2, Peters et al., 2005. The results suggest that more than one growth model could be required to best describe the growth function, the growth curve, and maturing patterns of various chicken genotypes. In another study, we used linear, exponential, and polynomial models in the prediction of live body weight from linear body measurements in pure local and pure exotic chickens and their crossbreds in southwestern Nigeria. A combination of shank length and breast guard tended to provide a better prediction of live body weight of chickens when compared to the use of breast guard alone. In another study, we compared 5 growth functions: the Brody function, the Gompertz function, the logistic function, the von Bertalanffy function, and the Richards function, to describe the growth rate and growth pattern of normal feathered local chickens, the naked neck local chickens, and the Marshall. The Gompertz, the von Bertalanffy, and the logistic functions fitted the growth patterns of all 3 chicken genotypes very well, and the R-squared values were all above 99.89%. And based on all comparison criteria, the logistic model gave the best fit for the body weight age relationship for the chicken genotypes. And the Brody function and Richards functions proved unhelpful in fitting the growth data of these genotypes in this study, going by the parameter estimates, the p-values, and the convergence criteria. In another study, we have moved on in this modeling to apply the Bayesian approach in the fitting of body weight age relationship. The Bayesian approach uses prior knowledge of previous estimates in the field and is relevant for the tropical environment, most especially where we have paucity of data. And using this Bayesian function, we recommend that the use of Bayesian weighted multiple regression model in fitting linear body measurements of local chickens in the body was better, as it fitted better than unweighted multiple regression models. A study there showed, as published in Adenike et al., 2019a. We have also developed sex-specific statistical tools that can successfully predict and differentiate Eimeria-infected and uninfected local chickens. Adenike et al. 2018a. Adenike is a student of mine that did his PhD in FUNAAB under our supervision and is presently a colleague in the Department of Animal Breeding and Genetics as a lecturer. We recommended that our 2 models below could be used to diagnose and predict chickens infected with Eimeria through routine blood tests and estimates of body weight gain, especially where key facilities and personnel are lacking. And we have that on the screen. For males, we have the one on top, the model on top, and for the females, we also have the one below showing the various parameters that are important, the PCV or the red blood cell or the weight gain at 3 weeks or white blood cell, all of them coming in there, and then the lymphocyte ratio as appropriate. In our study, we leveraged our understanding of the polymerase chain reaction techniques to attempt a molecular diagnosis of subclinical trypanosomal vivax infection in 3 breeds of goats in Nigeria. And this is another key area of study that one of our PhD students, Sonny, worked on for his PhD. Mr. Vice Chancellor, sir, I would like to go on to another area of my research, which was carried out at the famous Roslin Institute, Edinburgh, United Kingdom, under the sponsorship of the Commonwealth Scholarship Commission, which offered me a Commonwealth Fellowship in the year 2000. And part of the work we did was on the identification and mapping of quantitative trait loci for broiler chicken traits. And the Roslin Institute is the famous institute that cloned Dolly the sheep. Recent trends in livestock genetic approach have dwelt on the application of new technologies, such as the DNA marker technology, on animal improvement. The detection and mapping of quantitative trait loci, in bracket genes, on chromosomes constitute a key process for hastening the genetic improvement of animal performance, as the major genetic basis influencing the animal species is highlighted much sooner. Using an array of 101 microsatellite markers and an F2 population made up of 466 individuals drawn from 30 different families from a broiler line layer line cross, we detected and mapped quantitative trait loci, QTL, in chickens. Our research efforts have yielded widely acclaimed scientific reports on the quantitative trait loci affecting 3 of the most important productivity trait groups in chickens: growth, fat deposition and distribution, and meat yield. The quantitative trait locus we detected with the largest single additive genetic effect for growth in broiler birds was found in our study on chicken chromosome 4. And the effect of substituting 1 copy of this gene was an increase in the body weight of the bird, of the broiler bird, of 249 grams in the bird. We published this in the Poultry Science, and the one which is reputed to be the best poultry science journal in the world, and it's based in US. Sewala et al. 2002, and then another article published in Animal Genetics, another reputed foreign journal, IKEOBI et al. 2002. Our study found evidence of statistically significant QTLs on chromosomes 1, 2, 4, 7, and 8 affecting chicken body weight, and 1 statistically significant QTL on chromosome 13 affecting chicken weight at all 3 ages. Now, I talked earlier about chicken microchromosomes and macrochromosomes. I also talked about chicken sex chromosome Z and W. And the table that we have, table 1, shows the significant QTLs for chicken weight at 3, 6, and 9 weeks of age. At 3 weeks of age, we have QTLs, genes, you can call it, detected at the various respective positions in chromosome 1, chromosome 13, and the Z chromosome, which is the sex chromosome, at 3 weeks. And one of the interesting results obtained here is that at 6 weeks of age, the chromosome, the QTL that we detected on the Z chromosome at 3 weeks, statistically significant at 1% level, it diminished in effect and importance and approached significance at suggestive level. And that's why we highlighted that in yellow. I hope it's showing on the screen there, showing suggestive linkage at 6 weeks of age. What this is saying is that at the prenatal level, prenatal stage, an early stage of the life of the chicken, the Z chromosome is important. The gene on the Z chromosome controls the expression of the early stage, the weight, the expression of the live weight at the early life of the chicken at 3 weeks. It moves on, and at 9 weeks of age, that effect completely disappeared and was not detected at all, whether at significant level or at suggestive level at 9 weeks of age. There were highly significant and remarkable differences between the 2 sexes. We looked at this, and one thing this table is highlighting for us, oftentimes when people study fat laid down in chickens, they go for abdominal fat. I worked on abdominal fat for my Biography final year project at IFE in 1982, but we found out that the skin fat in the chicken is about twice the size of the abdominal fat, meaning that more some emphasis and more emphasis should be given on the skin fat. Now, when Nigerians consume broiler meat, they consume broiler meat along with the skin. They consume broiler meat along with the skin. And one thing I found out when I was in UK was that I had this flatmate of mine who was a European. And each time, you know, we cooked chicken with the skin. Before he ate it, he would remove the skin and eat only the muscle. But here in Nigeria, I think we eat everything. We eat both the muscle, we eat both the skin, and we eat both the what? The bone. What this is saying is that when we consume the chicken skin along with the meat and the muscle, we are consuming the chicken skin fat alongside. And this has importance. And you know, and I'm going to show you some models that has to do with comparative mapping and the various genes involved with respect to fat deposition in chickens. Now, with respect to abdominal fat, we have here on the table the chromosomes that control the expression of the abdominal fat in the chicken. And the interesting thing is that some of these chromosomes are the macro chromosomes, and some, like chromosome 28 and chromosome 7, are micro chromosomes, in line with our earlier submission that the micro chromosomes are richer in gene density. Of course, we have the expression, the chromosome that control the expression of the skin fat. Also highlighting this, there are micro chromosomes involved, more than macro chromosomes involved in the expression of the skin fat. Only one macro chromosome controls the expression of the skin fat, and 3 micro chromosomes control the expression of the skin fat in the broiler chicken, showing us that if we need to control the expression of fat in the chicken, we have to go we should be concerned with not only the macro chromosomes, but also the micro chromosomes. Of course, we have the chromosomes controlling the expression, the fat distribution in the chicken. Now, we went on to look at comparative fat QTL mapping results. And this particular result was presented in the 52nd European Association for Animal Production, EAAP, in Budapest, Hungary, in 2001, showing, and what this is showing, the first column has to do with the identified chicken chromosomes controlling the expression of chicken fat that we found in this study, whether abdominal fat or skin fat or fat distribution. And we also used comparative mapping techniques to identify the homologous mouse region or mouse regions and the homologous human chromosomal regions that are implicated. Each animal species, it must be understood, there are similar regions. If you take a human being, human genome, there are similar comparative genome or regions in the human genome that behave the same way behaviorally with the mouse genome, and of course with and like we found in this study, with the chicken genome. The interesting thing is that there are some candidate genes involved, such as the insulin-like growth factor 1 gene. We have the relevant authors that have found that. There are candidate genes such as propionmelanocortin, which is a candidate gene for skin fat, abdominal fat, and there are also genes such as the insulin-like growth factor 2, others like the serum leptin levels gene, and others such as insulin receptor genes. Now, what this is saying is that the broiler chicken can serve as a veritable, reliable model, okay, for the study and control of adiposity and obesity in both mouse and humans. So in the study, usually when you talk about adiposity, obesity, scholars, they dwell more using mouse, some use rats, and some go on to use humans. But we are saying here, based on our comparative mapping results obtained, which were published in literature, that the chicken can serve as a veritable model in the study and dissection of the genetic basis of adiposity and obesity. Now, with respect to the QTL work we did, we went on to look at QTLs for meat yield and muslin. And the interesting thing in this table, I just want to summarize it for the sake of time, is that there are different quantitative trait loci, chromosomal regions, and genes involved in the expression of different cut yields in the body, in the carcass of the chicken. There are different chromosomal regions that has to do with the thigh yield of the chicken. There are different chromosomal regions that have to do with the expression of the wing yield. There are different chromosomal regions that have to do with the expression of the thigh muscle yield. There are different chromosomal regions that have to do with the carcass, whole carcass, weight and breast muscle. So when we talk about genetic improvement, especially for meat production, we cannot be looking in a unidirectional manner. We have to look at it in an all-encompassing manner. And if you look at this table on the screen right now, there are both macro chromosomes and micro chromosomes, and of course the sex chromosome Z is implicated in the expression of the drum yield. So when you are eating the chicken drum, you need to know that the Z chromosome, which is a sex chromosome, is involved in the expression of the yield of the chicken drum. That's what this table is saying. A very interesting study here. Now, the chicken Z chromosome was found to harbor a quantitative trait locus, which affected live body weight at 3 weeks of age, and which suggestively affected live body weight at 6 weeks. I've said that. And IKEOBI et al., 2004, we published this in the Livestock Production Science, also found a QTL on chicken chromosome Z affecting drum yield and drum and thigh muscle yield, and also suggestively affecting wing yield in the chicken. And the remarkable results on the identification and mapping of quantitative trait loci for growth at 3, 6, and 9 weeks of age, abdominal fat deposition, skin fat deposition, fat distribution, and carcass yield, meat yield, and muslin in chickens have commanded a great deal of interest from several scientists, and have also received extensive and positive reviews and citations by several researchers such as Hawking et al. 2002, Hawking 2005, and Abashti et al. 2007. Vice Chancellor, sir. The next major plank of my research and contributions include our studies on genetic diversity and associated traits in Nigeria's indigenous animal resources. My research has integrated both DNA technologies with the classical animal breeding and genetic procedures in genetic diversity and association studies. Molecular level studies are specific because they give rise to detection of genes implicated in livestock productivity, adaptability, and livability of Nigeria's animal genetic resources. And several reports of our research activities and findings have been published in highly reputed international journals. And many postgraduate students, and indeed some undergraduate students, have been involved in these studies. So this has enhanced learning, it has promoted teaching, it has promoted research at that level, and their results have also been published as collaborative findings in highly reputed foreign journals. We've also looked at the evolutionary trends and relationships, and evaluated this for different livestock species and related it to what other people published in literature in foreign countries. Sequences of these genes have been deposited in the gene bank. This represents a major contribution from our research work, because sequences of these genes for the local breeds of livestock have been accepted. That is the sequences that we generated after sequencing of those genes, and I'm going to highlight them, have been deposited in the gene bank. The next table 6 shows that, And this represents the major scientific contribution. Now, if you look at this table, the first one was the tumor necrotic factor receptor superfamily 1A gene. And we worked on the Nigerian chicken and some exotic chickens. And if you look at the last column, it's talking about the gene bank accession numbers that have been given to us, issued to us. They accepted these sequences, deposited them in the gene bank, and these are available for consultation by other researchers, in different parts of the world. And this is important in the use for phylogenetic and phylogeographical studies to show the evolution of these genes and the evolution of these livestock species. We looked at also zyzine. Zyzine and the tumor necrotic factor receptor superfamily 1A gene are immune system genes that are very important in the survival of the animal for its productivity, adaptability, and livability in our local conditions. And the gene bank accession numbers have been also included. We looked at insulin-like growth factor 1 gene and toll-like receptor 4, toll-like receptor 2, and heat shock protein 90, and these have been shown and are shown on the screen. Using blood proteins as genetic markers, we carried out a preliminary screening of the genetic lineage of the different strains of the Nigerian local chicken. And the frizzled, naked neck, and normal feather chickens were clearly separate from each other, having a mean genetic similarity of 55%. The naked neck local chicken was the most divergent, Adeleke et al., 2011C. In another study, we compared the effectiveness of the random amplified polymorphic DNA, RAPD markers, with microsatellite markers in evaluating 3 local and 2 exotic chicken populations in Ogun State and Ondo states of Nigeria. The microsatellite markers gave a more accurate cluster than the RAPD markers, suggesting the superiority of the microsatellite markers in probing and eliciting the genetic background of the local and exotic chicken populations. One of our master students, who is presently on his PhD, Oni et al., 2016, worked on this. And using the PCR RFLP technique, we found that the effects of the growth hormone gene polymorphisms were significant for chicken body weight, blood weight, eviscerated weight, and breast weight. There were also significant sex by growth hormone gene polymorphism interaction effects on chicken carcass traits, Weto et al., 2016. Weto is a colleague in the department. We carried out a sequence analysis of the insulin-like growth factor 1 gene in 5 mammalian species, which revealed the presence of 5 haplotypes in pigs, 2 in cattle, 4 in water buffalo, and 1 in horses, Akifenwa et al., 2011. We extended this sequence analysis study to 4 poultry species, the chicken, the turkey, the duck, and the goose. And we found that the IGF-1 gene was highly conserved in the 4 poultry species, Obeto et al., 2011. Perhaps in line with this report of the conservation of the IGF-1 gene in the chickens, we found that the effects of the insulin-like growth factor 1 gene polymorphism were not significant for any of the chicken carcass traits we studied, Weto et al., 2017. In another study, we used 3 restriction enzymes, MSP1, BSCRI, and TAC1 to individually digest the PCR products of the major histocompatibility complex BL-beta 2 gene in 3 strains of the local chicken. Part of our work in FUNAAB has dwelt on the MHC region in the chicken, the major histocompatibility complex region, which is a region noted for immunity in the animal. The TAC1 enzyme exhibited monomorphic pattern with genotype AA and at a frequency of 1.0, perhaps suggesting the fixation of the naturally selected AA genotype of the BL-beta gene in the Nigerian local chicken strains, Akpan et al., 2019. Going further, like I mentioned earlier, in the table on gene bank accession, sequence accession numbers, analysis of the tumor necrotic factor receptor superfamily 1A and the zyzine genes in 3 local chicken strains and 1 exotic chicken strain, Nera Black, showed high nucleotide divergence and high haplotype diversity, which along with the evolutionary relationships suggested historically restricted gene flows among the chicken genotypes studied. Adenike et al. 2019b. The naked neck local chicken showed the highest level of genetic diversity in the zyzine locus. And these 2 genes are immune genes that protect the local chickens, the birds, against coccidiosis. The tumor necrotic factor receptor 1A gene is a multifunctional cytokine that facilitates the induction of cytokine secretion, thereby promoting host defense against intracellular pathogens and the activation of leukocytes, while the zyzine encodes proteins that are needed in wound healing and immune metastasis. We went on to report 6 SNPs, single nucleotide polymorphism. These are mutants, variants, in the heat shock protein 90AA1 gene in 2 exotic layer strains of chickens reared in Nigeria for commercial egg production. But association studies showed no significant effect on heat tolerance traits evaluated. One of our PhD students, my PhD student, Dr. Irioje, worked on this for his PhD, and his paper was published in a highly reputed international UK journal, Irioje et al. 2020. Now, this is a summary of some of the genes that we worked on and the polymorphisms we detected and the associated effects. Growth hormone gene, insulin-like growth factor 1, and of course the references of our publications are also included. The TNF RSF1A gene, the zyzine gene, the BL beta 2 gene, and of course the HSP90AA gene. The polymorphism detected are shown, and whether it is RFLP or single nucleotide polymorphisms, and the various associated effects are also highlighted here. With this, detected a novel polymorphism in the melanophilin gene in 3 Nigerian breeds of goats, which had no associated effects with coat color, as there were low differentiation within and among goat breeds in this particular locus studied, Adefenwa et al. 2013. I work on the exons 1 and 3 of the myostatin gene in the red Sokoto goat breed, focused on the amplification, sequencing, and bioinformatic analysis. A variant of the exon 1 region was significantly associated with body length of red Sokoto goats, which were extensively managed in our local environment. Sonny et al. 2019. My student Sonny worked on this for his PhD, and which he concluded in 2016. One of our students also, a PhD student, worked on tenascin gene with respect to shape, and he found that the polymorphisms, the SMP genotype has significant effect on body weight for tenascin. And tenascin is a particular gene that controls the expression of connective tissues in animals. And this, we found out, had effect on 4 cannon bone length in sheep and was significantly associated with pulse rate and skin temperature. Ajayi et al. 2013. And I would like to move on to talk about some of our students. Now, working with a Nigerian cattle breed, White Fulani, and a temperate breed, Angus, our study identified a previously uncharacterized gene called ODF1, owing to the presence of the intact alpha crystalline domain. Ajayi 2014. That's his PhD work found, and he got a fellowship, a scholarship from the Norman Borlaug Foundation to do part of his research in US, and his PhD work in FUNAAB, which he defended in 2014, was highly rated. We also investigated the toll-like receptor 1 and 2 gene polymorphisms, and this was important for trypanosomiasis traits in Muturu and White Fulani cattle. And when they were challenged with trypanosoma vivax, we found 16 single nucleotide polymorphisms, and which were detected, 7 in TLR1 gene and 9 in TLR2 gene. TLR is toll-like receptor, which is also part of the immune system genes in animals. Agbalaya et al., 2018. Agbalaya is a past PhD student of mine working in one of the higher institutions in Nigeria. And she's also here today. Now, this is a summary table showing the polymorphisms we detected in small ruminants that we have highlighted earlier, just like we highlighted for the chicken, and we have summarized the various references and associated effects in the species. We went on to look at leptin gene, and a postgraduate student of mine, Ogunwa, worked on this, and the leptin gene controls the expression of hunger and appetite in man and animals. And we found that the evolutionary relationships among 3 cattle breeds showed a separate clustering of the White Fulani cattle as a group, while the Kuri cattle and the Ndama cattle formed separate cluster, perhaps emphasizing the zebu nature and the taurine nature of these 2 different groups of cattle. Well, this is the summary table for the polymorphisms we detected in cattle, and then the associated effects of these genes with respect to the animals. The second to the last column shows the associated effects with effects on blood parameters, adaptability traits of body temperature, and some other productivity traits. And these genes span the toll-like receptor family genes, the leptin, the heat shock protein genes, which act as molecular chaperones that help the animal to fight thermal assaults and withstand heat stress, especially such as is prevailing in our environment. We also looked at HSP90 gene polymorphisms in 4 Nigerian zebu cattle breeds, and we found that polymorphisms existed at position 116, 220, nucleotide position 346, and 390. And these 4 breeds of cattle were worked upon, and these are Nigerian breeds: Red Bororo, Sokoto Gudali, Ambala, White Fulani. And these are the various, and some of them have effect on the amino acid change. And when there is an effect on amino acid change, such as we are highlighting on the screen, it will affect protein function. And of course, it will also affect the performance of the animal in various ways. These are details of some of the polymorphisms found, details of whether it is in terms of transition, indel, or transversion. And these are the cattle breeds we worked upon: Ambala, the White Fulani, the Sokoto Gudali, and the Red Bororo breed. Now, part of our studies was on expression, gene expression, and specifically the expression of the BL-beta-2 gene at cytolytic and latent immune response stages in Nigerian indigenous local chickens. And what this table is showing is that different organs of the chicken express this gene, and it's an immune system gene in different ways. And the spleen, for example, has higher expression, both at the cytolytic and latent stages, than the thymus. And then if the bird is a high titer bird, it has a higher expression than low titer birds. And then the male chicken has higher expression of this immune system gene than the female chicken. And among the major genes we worked on, there were 2 differences. in the expression of this gene. We worked on the kinetics of the interleukin-10 gene polymorphisms and their effects on body weight, and the study showed that the AA genotype of the interleukin-10 gene had a higher expression in response than the AB genotype. That's what this table is saying. And of course, the genetic line also depends, it also controls whether it's going to be high expression or low expression. And then the sex of the chicken also has to do when it is combined as an interaction with titer value, whether it will affect the expression or not, like this table is showing so. And then we also looked at attenuated salmonella, and as part of our study, we are presently, with respect to the Nigerian local chicken, we have gone to the 6th filial generation. And at each generation, we evaluate the performance of the chicken, not only phenotypically, but also at molecular genetic level. And this has highlighted some of the important results we have been emphasizing. These are some of the chickens that we are working on at the FC levels now. Summary. Apart from our research on the mapping and quantitative trait loci for performance traits and the various works done on other livestock species, as reported earlier in this lecture, our work on the Nigerian local chicken can be summarized under these following headings, and we have them from the first to the last one, which is number 27. The details are available. But they all encompass not only the phenotypic the characterization, quantitative genetic characterization, but also molecular genetic characterization and the identification and mapping of QTLs and gene expression studies at the molecular level. The successes we have registered in this department over the years emphasize: 1, poultry selection and improvement, special recognition as a reference point in our department on work on the local chicken. Scientists and researchers come from afar to obtain foundation stock on local chickens for their own research. And the Department of Animal Breeding and Genetics is the very first in the Nigerian the very first Department of Animal Breeding and Genetics in the Nigerian university system. And the department has the richest assemblage of animal breeders and geneticists among all universities and institutions in Nigeria. The department produces, of course, the highest number of PhD degrees in animal breeding and genetics each year compared to any other university in Nigeria, and successful integration of molecular genetic approach in the teaching and research in animal breeding and genetics, and attraction of patronage of our postgraduate programs from graduates of other universities in Nigeria. Suggestion, sir. There is the need for the conservation of our indigenous animal genetic resources in Nigeria. There is also the need for the establishment of multiplication centers for the indigenous animal genetic resources with emphasis on genetic purity and breed distinctiveness. There is also the need for a full, comprehensive, and up-to-date livestock census in Nigeria. We cannot continue to rely on outdated data on animal populations in Nigeria. There is the need for increased funding by relevant agencies for research in universities. Eateries, caterers, food vendors, and domestic consumers should always process the chicken skin and underlying fat before cooking, roasting, or grilling, so as to enhance the quality of chicken meat and minimize the intake of chicken fat by consumers. Our work has also provided research evidence that chickens can serve as model in our concerted efforts to dissect the genetic basis of adiposity and obesity and associated conditions in humans. We commend the government for the establishment of a distinct department of animal husbandry services at the federal level to coordinate the formulation and implementation of policies geared towards the development of animal agriculture subsector in Nigeria. The creation of new research institutes to handle research on specific animal species is long overdue to ensure that targeted attention is focused on each animal species and to improve their contributions to the food and nutritional needs of Nigerians. There is the need to call for a lasting solution to the incessant cattle herders, crop farmers clashes in different parts of the country, and to end the carnage and displacement of persons that have been the results of such clashes. Appreciations. I would like to sincerely thank the Vice Chancellor of this great university, Professor Felix Kolawole Salako, for continuing to create the enabling environment under which FUNAAB has continued to thrive and to excel. Thank you very much, sir. I would like to thank former Vice Chancellors of this university for their immense contributions to the development of FUNAAB. They are Professor Eno Adedipe, Emeritus Professor J.A. Okoje, late Emeritus Professor I.F. Adu, Professor Ishola Adamson, Professor O.O. Balogun, Professor O.B. Oyewole, and Professor O.A. Eniko Mehi. I would also like to thank this great university that has offered me the opportunity to serve as a staff right from 1990 to date, and for the opportunities this university has offered to me in terms of administration, being part of the administration at the departmental, college, and student affairs level, and even at council level. I am indeed grateful. I would like to thank my academic supervisors, Professor J.O. Akiokun at Ife and late Professor L.O. Ngere at Ibadan for their immense contributions to my life and career. I want to also recognize that one of my former teachers, lecturers at Ife, is here today, Professor Abisogun Lee, a former Vice Chancellor of the Lagos State University, LASU, Ojo. Thank you very much, Professor Lee. Thank you, my former teacher at Ife. Thank you, sir. You're welcome. I'd like also to thank, I'm grateful to Professor J.A. Oliemi and Professor G.N. Ebunike of the University of Ibadan, who as heads of departments offered and retained me as teaching assistant to enable me conclude my PhD in 1990. I'm grateful to the following agencies: the Commonwealth Scholarship Commission in the United Kingdom that offered me the Commonwealth Fellowship in 1990, Roslin Institute, Roslin, Scotland, that offered me a place as a visiting scientist, in 2000, not 1990, 2000. International Livestock Research Institute, Kenya, Nairobi, Kenya, Canadian International Development Agency, that have supported my work and some of my travels, International Development Research Center, Canada, ISGEB, International Center for Genetic Engineering and Biotechnology, and the Technical Center for Agricultural and Rural Cooperation, CTA, Netherlands. I'd like to thank the TED Fund, Tertiary Education Trust Fund, Abuja, that funded some of our research work. I would like to thank my foreign collaborators and supervisors, Professor John A. Williams, Professor Hawking, Professor Chris Haley, Professor Dave Bott, and Professor John Gibson. I also thank the Cornell University and its staff, Dr. Ikide Imomorin, for his collaboration with our work in the department. I would like to thank and appreciate Professor Mrs. O.A. Adebambo, who has been our mentor over the years in the Department of Animal Breeding and Genetics. She has not only been our mentor, she has been our mother, and she has been supportive of and she has been in the forefront of the work we have been doing in the department. I pray that the Lord will continue to keep us strong and healthy in Jesus' name. My colleagues in the department are very much appreciated. They are Professor Zoje, Professor Bemji, Professor A.O. Adebambo, Dr. B.M. Ilori, my HOD, Dr. M. Weto, Dr. Adena Ike, Dr. Duro Sharo, Dr. A.J. Sonda, and Dr. Yuapan. Along the way, two of our colleagues passed on to glory, Dr. Mrs. J.A. Adenowo, and Professor O. Olowofeso. We pay tribute to them on this occasion. Some of our colleagues have moved on to greener pastures elsewhere, and they are Dr. S.O. Peters, Dr. M.A. Adeleke, Dr. V.E. Olori, Dr. R.A. Lawal, and Mr. I.O. Shomi. We remember them today. The other staff of our department are very much appreciated. Mr. Femi Adesoya, Ms. Bola Odedairo of blessed memory, Mr. O.A. Awoyemi, Mrs. C.J.N. Shodelola, Mrs. O.R. Shaba, and Mrs. S.A. Abiodun. I thank them all. The staff I worked with as head of department, as Dean Student Affairs, and as Dean Kolanem are also appreciated. Some of them are Mrs. K.A. Babatope, Mrs. Shonde, Mr. A.C. Chime, Mrs. B.O. Akitunde, Mr. Shegun Oguyemi, Mr. Tobi Ogunsheye, Mr. Edu, Mrs. M.A. Bileu, Mrs. C.G. Oduto, Mrs. Obembe, Mrs. Shweto Yusuf, Mr. A.E. Akintayo, and Mr. Mike Ogunji. Professor Chris Onwuka and Professor John Singario are appreciated for their contributions to my career, especially during the early years. The following friends of mine are appreciated: Professor Nnamdi Ezeri, Professor Felix Mafiana, Professor and Professor Mrs. I.C. Eromosele, Canon Professor Ore Ayelagbe, Professor Wilfred Alegbeleye, Professor Olusegun Mark Idowu, Professor Ikechukwu James, Dr. Israel Oshushino, Professor Fred Smith, Dr. Julius Williams, Dr. and Dr. Mrs. J.A. Abiono, Dr. Richard Shobayo, Professor Samuel Sanwobo, Professor Alabajola Osho, Professor Mark Shogule, Professor Moses Adedire of blessed memory, Professor O.G.F. Nwogo, Mr. Madubuko, Mr. Ibuna, Mr. Madoka, and Mr. Ibeto. Members and secretarial staff of Senate Coordinating Committee on Examination Results, SECOSA, are very much appreciated. From my experience, they are the best members of any university committee in this university. I commend them all. I want to appreciate the president of the Nigerian Institute of Animal Science, Professor B.Y. Abubakar, and the registrar of the Nigerian Institute of Animal Science, Professor E.A. Iyaye, and the council of the institute for the recognitions that the institute has given me over the years. I would like also to appreciate the president of the Animal Science Association of Nigeria, an industry practitioner, Mr. Raymond Isiadinso, for again the National Executive Committee of ASAN for the recognitions that I have been given by the association. Mr. Vice Chancellor Star, I would like to appreciate this friend of mine, and this inaugural lecture has been written long before now. Professor Oluseyi Oluwajubelo Oluwatosi, a friend of mine that has worked with me over the years. And he and his wife, Professor Mrs. Bamidele Oduguwa, they have sacrificed in more ways than one to take care of me and my family in unusual ways. They have worked alongside with me over the years in the Chapel of Grace, and I remember him on this occasion. Professor Oluwatosi passed on to glory yesterday. I would like to appreciate these members of the Chapel of Grace, Federal University of Agriculture, Bekota, who have been very helpful, motivating, and inspiring in serving the Lord with me through the years. Dr. and Mrs. A.O. Fafiolu, Professor and Mrs. T.O. Fabumi, Dr. and Dr. Mrs. Ayo Ajasa, Pastor and Mrs. Chike Zebazo, Professor and Professor Mrs. A.O. Dipeolu, Dr. and Mrs. Y.I. Irioje, Dr. and Mrs. M. Weto, Dr. Alaba Dare, Mr. and Mrs. Ayo Famugwele, Mr. and Mrs. Omotosho Ogumola, Mr. and Mrs. Adeleke Adekombi, Mr. and Mrs. Abiodun Moshako, Mr. and Mrs. Yinka Aluko, and a host of others for their numerous contributions. God bless them both all. The members of the Chapel of Grace alumni group, some of whom are here, are very much appreciated. The president of the Chapel alumni group is here, Engineer Lanre Fabi, and the others, they appreciated. Dr. and Mrs. Anselm Diaye, Engineer Ife Ajibola, Sheyi Ambiodu Joseph Umvenu, Pastor Wale Onodeko, Dr. Mrs. Tolu Adeleye, and Mrs. Cindy Opeyemi. These friends of mine, some of whom are here today, are also appreciated. Evangelist Wala Kombi, Venerable Alfred Oreduba, who is here from Lagos, Bishop Jide Orire, who sent a representative from Ibadan, Venerable Samuel Onofeso, Most Reverend Nicholas Oko, Bishop Richard Ige, who is here today, late Reverend Dr. Gomba Oyo, Apostle Lawrence Achudume, Professor Silas Egbonwo of LASU, late Professor Ikechukwe Zago of UNN, Apostle Julius Oluwafemi, Dr. Ayobami Oyedele of OAU Leife, Dr. John Ihediohama, Dr. Bayo Adaramola, Professor Louis Nwokocha of UI, Reverend Professor Obiora Nwosu of UNIZIC, Professor Joseph Mwojeku of UNILAG, Engineer Biodu Fijabi, Reverend Joseph Idemo, who is here today, Dr. Sio Diokoronkwo, Venerable Obiora Anyika, Pastor Mike Roberts, and Pastor Titus Lambo. The Student Christian Movement of Nigeria is very much appreciated. This was my Christian fellowship on campus, first degree, second degree. They took care of me in several ways, and I pay tribute to them today. I remember them with fondness. The Full Gospel Businessmen's Fellowship International, Abeokuta, that's where I met my wife. They are very much appreciated. Professor and Dr. Mrs. Sam Oluwalano, who is one of the leaders, and others such as Dr. and Professor Mrs. Enyolorunda. My past PhD students are here. Some of them are here, and they are very much appreciated. Professor C.A. Chineke of FUTA Akure, Professor A.O. Adebambo of FUNAAB, Dr. Amusho, Dr. Ajayi, US, Dr. Ogunshola, US, Dr. Mrs. Bassi, Dr. M.T. Sonny, Dr. Mrs. Agbalaya, Dr. Irioje, Dr. Akpan, FUNAAB, Dr. G.O. Onosoya, Dutse, Dr. J.S. De Campos, Unilorin, Dr. D.O. Ogunlaki is here today, and Dr. A.S. Adenaike deserves special mention, and he knows why. My current PhD students, Mrs. Emo Sonda, Mrs. A.J. Akimbinu, Mr. O.A. Oni, and Mr. J.O. Wanodi, a lecturer in Port Harcourt, who is also here today, are appreciated. My past PhD master students are appreciated, the list is there. And the parents of some of my postgraduate students are appreciated. Mrs. M.O. Ajayi, Otumba Onosoya, Mrs. F.A. Akifenwa, Dr. Mrs. De Campos of blessed memory, and Professor Fajemisi of FUTA. Mr. Vice Chancellor, sir, I want to appreciate my loving parents, Sir Dennis Chukwemeka and Lady Virginia Akoma IKEOBI, both of blessed memory, for their immense contributions in my life. They contributed not only grammatically, they contributed genetically to what we are seeing today. And in addition to the grammatic and genetic contributions, they have contributed financially, materially, morally, socially, and emotionally to what we are witnessing today. On this occasion, I remember them because they first taught me to work and to work hard. And over the years, they kept me at it. I remember my loving parents on this occasion. I would like also to thank my uncle and auntie, Sir Isaiah Onyekozulu, and Lady Deborah Ijoma IKEOBI for their immense contributions to my life. My uncle, Sir Isaiah IKEOBI, he edited the Holderness and Lambert book in chemistry, and he taught for several years at International School, University of Ibadan. I remember him on this occasion. He's 90 this year, and he's not able to come, but he has told me to reserve a copy of my inaugural lecture for him. I think he's going to examine me again, whether it measures up to his own writings too. I would like to thank my only sister, Pastor Mrs. Njireka Edith Nwafo, the only girl in a family of 6 children. And sometimes his 5 brothers have not been cooperative with her. She has been the dutiful, loving, and caregiver in the family. She's not able to come. She's based in Port Harcourt, but I remember her and I thank her for her contributions to my life. I want to thank my elder brother, again of blessed memory, Venerable Dr. Chukwudi Ernest IKEOBI. And here we are together in this picture. My brother, I want to thank God for his life, because over the years, his shoes have remained too big for me to fill. I want to thank my other brothers, Victor Udechukwu IKEOBI, Pastor Kingsley Chiedu IKEOBI, and Pastor Emeka IKEOBI. We say, I come from a family of pastors. Mr. Vice Chancellor, sir, I came to Abeokuta in 1990, and a family accepted me readily and wholeheartedly. And they were willing to give me their cherished princess as my wife. I remember today my parents-in-law, Chief and Chief Mrs. Isaac Olusoya Babarisa, both of blessed memory, for their unconditional love for me. Of course, their daughters are here with their husbands. I also remember my wife's auntie, Mrs. Abiodu Peters, of blessed memory, for her love and care for her family before she passed on. I appreciate my wife's cousin, Mrs. Agnes Ogunremi, for her care for her family through the years. And I appreciate my wife's siblings, Tolu in London with his family, Dupe here, she's here today with her husband, and Yemisi in the US, and Shehu here in Abeokuta, and Yemisi in Abuja, Tayo in US. Please, I beg you, don't withdraw my wife from me. But I thank you all. Here is the picture of my father-in-law with my biological mother-in-law in the middle. I never knew her, I never met her. And then my stepmother-in-law that I know so well. But all of them are of blessed memories. I would like to appreciate my children that are here. And I would like to appreciate my children. They are Chinenye is there. Chinenye, stand up and take a bow. Precious, now Mrs. Udaja, she's heavy with child in Anambra and she cannot come. Uchechukwu is there. Uchechukwu, take a bow. Goodness, and of course Samuel. Samuel is not available today. I want to also say that 4 of these children of mine, there are 5, 4 of them are graduates of this great university. I believe so much in this university that I never wanted any of them to go to another university. One of my children actually filled in to go to IFE because she wanted to test what I tested at IFE. But at the last minute, I didn't tell her. She was a student of FUNAAB at the time. I shifted her application, change of university form to FUNAAB. And she got ready, first batch admission in FUNAAB. She's a graduate of FUNAAB today, Uchechukwu. And I want also to let them know that I'm very proud of them. And I couldn't have asked for a better set of children. They are the best 5 in a million. Here they are. And here we are. To my wife of many years and still counting, Lady Elizabeth Olubanke IKEOBI. This is a wonderful opportunity to say thank you. I appreciate my wife's positive and immense contributions to my life and career. She has been loving, kind, supportive, prayerful, diligent, companionable, friendly, self-effacing, and humorous. And I cherish every moment of these years she has shared with me. I want her to know again and again, today and always, that I love her with all of my heart, today and always. I thank her for everything she has done to take care of our family. If you like, this is a testimony, and there is a small boy of 6 months offering you a bouquet of flowers on this occasion today, and that small boy is standing before you today. Finally, on this occasion, I would like to tell about a journey I made across the Niger some years ago. In September 1977, I boarded one of the luxury buses at Onitsha on my way to Ile-Ife in quest of the proverbial golden fleece. I had with me just a suitcase and a small bag with just about everything I had in those days in them. I got to Ile-Ife and God turned my life around with an undeniable and unmistakable spiritual regeneration experience along the way. He saw me through the inimitable and the giant humbling Great Ife without a carryover, which was unusual in those days. Today, I want to thank God for his blessings upon my life. Next, I went to Ibadan against all odds, thinking that Ibadan was going to be a much friendlier place, a walkover, if you like. Well, in spite of the great odds I faced at Ibadan, God saw me through successfully. Today, I want to thank God for his blessings upon my life. After Ibadan, I came to Abeokuta, and God has seen me through all the changing scenes of life up to this moment in time with uncountable blessings that I could never have imagined. Today, I want to thank God for his blessings upon my life. He is worthy of all my praises. And so, on this occasion, this is my song of appreciation to the Almighty God. Then sings my soul, my Savior God, to thee. How great thou art, how great thou art. Then sings my soul, my Savior God, to thee. How great thou art, how great thou art. Mr. Vice Chancellor, sir, thank you very much indeed. May God bless you all.
Thank you, Professor IKEOBI, for a wonderful lecture. I will stop eating the skin. I will stop eating the skin of fowls now. The university is presenting this award of honor to Professor Christian Obiora Ndubuisi IKEOBI as the 62nd Inaugural Lecturer of the university. Congratulations, sir. The inaugural lecturer has actually recognized some of our guests, but I want to thank everybody on behalf of the university for being here. Our staff, academic and non-teaching, our students, and every other guest who is here. I want to specifically thank Lady Elizabeth IKEOBI, the wife of the inaugural lecturer. He has said so much about you that I do not need to add any other thing. Thank you, ma'am. Also, the daughters who are here, Chineye IKEOBI, you are welcome. Uchechukwu IKEOBI, you are welcome. I want to thank on behalf of the University Bishop Richard Ige, presiding Bishop, Covenant Library, Covenant Litany Church. You are welcome. Chief Lai Labode, Baronyo of Egbaland. You are welcome, sir. Professor A.B. Dou, Vice Chancellor Samuel Adegboyega University. Here, and remember you are still a staff. Professor Abisogun Olubode Lee, former Vice Chancellor LASU. You are welcome, sir. We appreciate you. Dr. Folukemi Akinusi, Chairman, Animal Science Association of Nigeria. Venerable Kayode Orejuba, Anglican Church, Lagos. You are welcome. Mrs. Tosin Olaogun and Mrs. Shion Ogudari, sisters-in-law. Welcome, madam. Mr. Kunle Ogudari, in-law to Professor IKEOBI. Reverend Joseph Edemo, General Overseer, Fingers of God, Covenant Church. Welcome, sir. Dr. Mrs. K.K. Agbalaya, animal scientist. Mr. Oluwole Ajayi and Mr. Larry Fabi, friends of Professor IKEOBI. Thank you very much. I think I should do something I didn't want to start with, more so when Professor IKEOBI mentioned it. Let us try for a minute silence for Professor Oluwatosin, who passed away yesterday. May his gentle soul rest in perfect peace. Please sit up.
Please, let's give a round of applause to our Vice Chancellor. We are on the verge of drawing the curtain of the 62nd Inaugural Lecture of our university. But you will agree with me that our teachers' rewards are no longer in heaven. That is why, within a short space of time, we want to quickly take the presentation to our inaugural lecturer from his former students and, of course, the Boys Brigade. They are going to do it both physically and genetically. Apology to the inaugural lecturer. So the boys, we are starting with the former students. Please, let's make it as brief as brevity. Wherever you are right now, please come forward. Time is actually not on our side. Former student, please. Please, let's give them a round of applause as they are coming to do it.
On behalf of the past students and the present students, we present this gift to our great leader. Thank you, sir.
Thank you very much. Thank you very much. Thank you very much. God bless you.
Thank you so much. The Boys Brigade, where are you? Let's have you quickly, please. Boys Brigade, let's make it snappy.
On behalf of the Boys Brigade, 2nd Ogun Cadets Company, hereby present this. Second General Salute.
Thank you so much. Those gifts are not winking. Well, distinguished ladies and gentlemen, this is where we have to draw the curtain of our 62nd inaugural lecture. We give all the glory to God and we thank you so much. Right now, quickly, we take the university anthem. Then thereafter, we take the national anthem. Let's all rise for the university anthem. Thank you so much. National anthem. Thank you so much. Thank you. Please, I want us to actually give honor to whom honor is due. Let's allow our presiding officers here on this side of the hall to file out first as a mark of respect to them. The exit points for dignitaries on this side will be on my right-hand side. While they are doing so, others will remain standing as a mark of respect to our principal officers and, of course, the 62nd inaugural lecturer. Thank you so much. Our Vice Chancellor, Professor Felix Kolawole Selako. We appreciate you, Professor. The DVC Development, we appreciate you, the inaugural lecturer. The DVC Academics, Professor Mrs. Bonali Akerudu, we appreciate you. The registrar, Dr. Bola Adekola. The bossa, Mr. Chukwuemeka Ezekpiasu. And of course, the University Librarian, Dr. Ms. Feyintola Onifade. The Dean of Copeland, Professor Murphy Atayeshe. And of course, the Dean of Kolanim, Professor Olusi Showande. We appreciate you all. The academic, Bishop Ige, we appreciate you. Thanks for coming. Professor Abisogun Lee, we appreciate you. And all other eminent guests that came and that grace today's second inaugural lecturer. Thank you so much. All my colleagues in public relations, the ceremonial committee, the chairman ceremonial committee, all the ceremonial committee members, the pressmen, we appreciate you so much. FUNAAB Radio, we appreciate FUNAAB Radio, we appreciate public relations and everybody, every group, every well-wisher that came for this 62nd inaugural lecture. Thank you all. For those of you that are traveling back home, we wish you a journey mercy. Thank you, ICT. Thank you, the decoration. Thank you, the Boys Brigade. Thank you, Manowa. Thank you, everybody. Thank you all. Thank you so much.